When you think about huge scientific breakthroughs, you probably picture complex experiments on, well, complex creatures. But what if I told you that the actual rule book for all of life, the very foundation of modern genetics, was written by the absolute smallest, simplest things on our planet? Today, we're going to dive into how these tiny titans completely unlocked the secrets of our own DNA. That's right, we're talking about bacteria and the viruses that hunt them down. For decades, these guys have been the real unsung heroes in the story of genetics. They were the perfect little biological test tubes that helped us to code the very essence of life itself. So this is the big question for today. What did these microscopic critters really teach us? And the answer, you might be surprised to hear, is pretty much everything that mattered in the early days of genetics. No exaggeration. I mean, just look at this list. It is absolutely wild how fundamental these discoveries are. We're not talking about some minor details here. This is the bedrock of the entire field. The very first proof that DNA was the stuff of life? Check. The definition of a gene? Yup. How genes talk to each other? You got it. All of this came from studying these tiny organisms. Okay, so to really get how this all happened, we need to meet the main characters in this microscopic drama. On one side, you've got your bacteria, and on the other, you've got a special type of virus that infects them, known as a bacterial phage, or, just phage for short. Now, not all phages are created equal. You've got guys like the T4 phage. Think of it as a brute force attacker. It's got one mission and one mission only: get in, multiply, and destroy. But then you have something like the lambda phage. It's more of a cunning strategist. Its genome is smaller, more nimble, and it gives it options. It can either go for the quick kill, or it can play the long game. And that choice is everything. So let's stick with that clever lambda-phage for a second. When it latches onto a bacterial cell, it has to make a crucial decision. And that choice leads to one of two totally different outcomes. Pass number one is what we call the lytic cycle. You can just think of it as a hostile takeover. The phage injects its DNA, and that DNA immediately hijacks the cell's entire operation. The poor bacterium is forced to stop whatever it was doing and turn into a factory, churning out hundreds of new viruses. And then the final step, the cell literally bursts open. It lyses, releasing this new viral army. It's fast, it's brutal, and it's incredibly effective. But then there's path number two, the lysogenic cycle. This is the sleeper agent strategy. The phage injects its DNA, but instead of going on the attack, it does something sneaky. It weaves its own DNA right into the host cell's chromosome. It just hides there, completely dormant. Every time the bacterium divides, the viral DNA gets copied right along with it, spreading silently to new generations. It's an amazing way to spread. Until some kind of trigger, maybe the cell gets stressed, flips a switch, and boom, it pops out and activates that destructive lytic cycle. And this table just lays it all out perfectly. The lytic cycle? The cell is destroyed. Fast. The viral DNA stays separate, and you get a massive, immediate viral burst. The lysogenic cycle? The host cell gets to live, for a while. The viral DNA actually integrates itself, and the whole game is about slow, stealthy spread. Two totally different strategies, all from the same virus. By the way, there's a cool name for that hidden piece of viral DNA. It's called a prophage. It's the virus in its quiet, integrated state, just lying in wait. And this is a really, really important concept. A piece of foreign DNA has just become a permanent part of the bacterium's genetic code. I mean, this is basically natural genetic engineering happening all the time. And that idea becomes a huge deal later on. Right, so this whole idea of DNA moving between organisms is a massive theme here. And it's not just something viruses do. Bacteria themselves have all sorts of ways of sharing genetic information with each other. It's kind of like their version of sex. There are three main ways they do it. First up, you've got transformation. This is probably the most passive way. A bacterium can just absorb little bits of stray DNA that are floating around in its environment. Seriously. It's like finding a random recipe page on the street and deciding, hey, I'm going to add this to my cookbook. It's a bit random, but sometimes you pick up a really useful new trick. Then you've got conjugation, which is way more direct and intentional. This is the closest bacteria get to actually mating. One bacterium literally builds a tiny little bridge to another one and actively passes a copy of its DNA across. So unlike transformation, this is a deliberate transfer of information from one cell to another. And third, we have transduction, which brings us right back to our phages. This is what happens when a virus is assembling itself inside a host and accidentally packages up a piece of the host's DNA instead of its own. Then, when it goes to infect the next cell, it injects that bacterial DNA instead. The virus becomes this unwitting little delivery shuttle, moving genes from one bacterium to another. It's what connects this whole world of genetic sharing. So as you can probably tell, there's this incredible, dynamic world of genetic information flying all over the place. And it's all part of a much bigger story. This constant, multi-billion-year-old arms race between viruses and the cells they want to infect. Viruses evolve to attack, and hosts evolve to defend. Back and forth, forever. And over all that time, viruses have developed some truly mind-bending tricks to stay in the game. They can constantly change their coats, their surface proteins, so our immune system doesn't recognize them. That's what the flu virus does. They can use latency, just like that lysogenic cycle, to hide out inside our cells for years, which is the herpes virus's whole deal. And some, like HIV, are even more aggressive. They make proteins that actively shut down our immune response. It is a relentless game of cat and mouse. And that all brings us to one last really powerful thought. For billions of years, viruses have been the planet's most successful, most prolific gene editors. They've been cutting and pasting DNA forever. And by studying their strategies, we didn't just discover the foundations of genetics, we were inspired to create our own revolutionary gene editing tools like CRISPR. So the real question isn't just what they've already taught us, but what incredible secrets are they still holding for our future?